But sure that we will mention that.
All right, good morning, everybody. First of all, I'd like to thank Sandeep, Martin, Anna, and Katia for organising such a fabulous meeting so far. Hopefully, I'm not going to screw that up. I'd also like to thank Agnes Enyedi for the kind invitation to present during this session. And I'd also like to second that it's great to see John Penniston here in the audience as well, who is a pioneer in the PMCA field. So today I'm going to be talking about two diseases, pancreatitis and pancreatic cancer. These diseases are both inextricably linked by cellular metabolism and the control of resting calcium. And of course, central to that is the PMCA. This is, okay, it's not working. Oh. Central to that is the PMCA, which is an ATP-driven calcium pump, which is critical for maintaining low resting calcium. And in particular, in these cells, these are non-excitable cells, pancreatic asthma cells and pancreatic cancer cells, PMCA is the last gatekeeper for the control of resting calcium because there's no sodium-calcium exchange expressed. And so cutting off this ATP supply from the PMCA inevitably leads to a cytotoxic calcium overload that leads to cell death. And this is the hallmark of many diseases around the body, but particularly pancreatitis. And in pancreatic cancer, glycolytic ATP is specifically involved in fueling the PMCA, which gives rise to apoptosis resistance. So what I'm going to be talking about today is I'm going to show you some very preliminary data showing the next generation of peptide inhibitors of the PMCA, which we believe may represent novel antimetastatic drugs. I'm also going to show you that cutting off the glycolytic ATP supply to the PMCA in pancreatic cancer might be another therapeutic strategy for this disease. I'm also going to show you how endogenous release of insulin from pancreatic beta cells within the pancreas has a direct protective effect on pancreatic asthma cells during injury, during acute pancreatitis. And this might also be something that can be exploited therapeutically. Okay, so onto cancer. Pancreatic ductal adenocarcinoma has very grim survival statistics, and this is particularly striking when you compare it to breast cancer, which incidentally receives about 15 times as much grant funding. Just saying, just in case you didn't know. Pancreatic cancer is highly resistant to most chemotherapeutic drugs, which only extend survival by a few months. However, there is some hope, and there are three strands of research that I would argue are very important to tackle this disease. The first one is early detection. So development of new biomarkers will hopefully help this because pancreatic cancer is detected very late in the course of the disease. Often patients don't get any symptoms until the disease has metastasized to other organs. Pancreatic cancer is also a highly desmoplastic tumor. It's very dense, the blood vessels are closed, and this makes it very difficult to deliver any cytotoxic drugs to the tumor. So targeting the tumor microenvironment is an important strategy if we're going to tackle this disease. But even with these two important strategies, the development of novel drugs that specifically target pancreatic cancer cells and either kill them or stop them from spreading must remain a central research strategy, and that's what I'm going to talk about. So this is a diagram taken from Hanahan and Weinberg's seminal review article explaining the hallmarks of cancer. Importantly, calcium signaling is involved in every single one of these different cancer hallmark responses. Yet, currently, there are no drugs that target the calcium signaling machinery for the treatment of cancer, which might seem surprising, although Greg Monteith's talk yesterday shows some promise. And this is highlighted in a review article that I wrote with my former postdoc, Andrew James, a few years ago, where we kind of summarized and highlighted some of the key targets of the calcium signaling machinery for potential treatment of cancer. And the reason I'm showing you this is that we raised a question in this article, and the question is, does the calcium signaling machinery offer good therapeutic targets for cancer treatment? And really, this will depend on whether the target is ubiquitously expressed or exhibits widespread function, in which case, any specific inhibitor may well exhibit adverse effects. Whereas if the target has unique expression in the pancreatic cancer or in the cancer cells or a unique function, then that's likely to give rise to a good therapeutic target. And what I hope to convince you, albeit with very preliminary data, is that PMCA4 might represent one of these. And the reason for this is that PMCA4 appears to be almost exclusively expressed in pancreatic cancer cell lines, such as myopathy cells, but also BXPC3. And in human tumors, PMCA4 is overexpressed, and this leads to poor patient survival. It's important to note that PMCA1 and PMCA4 are both ubiquitously expressed around the body. So targeting PMCA4 specifically means that it's unlikely that there will be adverse effects, because PMCA1 may give rise to compensatory mechanisms. And therefore, the unique expression of PMCA4 in pancreatic cancer cells may offer a really good therapeutic target. So before I go any further, I want to explain how we measure PMCA function in intact cells, which is really important. So we can inhibit the circuit pump with cyclopyrazonic acid, which depletes the ER of calcium and activates store-operated calcium entry. This flood of calcium into the cytosol means that the PMCA is now working at full tilt. If we now remove the extracellular calcium, we can monitor the cytosolic calcium clearance rate, which is directly proportional to PMCA activity. And this is a really important way of measuring functional PMCA in intact cells. We can apply paired experimental design and compare the rates, and in time-matched control cells, this is around about 100%. If we apply various treatments, drugs, whatever, we can then compare the effect of that drug on PMCA function. It's a really important way of measuring PMCA. One of the problems that has plagued the PMCA field is a lack of good inhibitors of the PMCA. Millimolar lanthanum is very effective, but of course it binds to every calcium binding site, so it's non-selective. Carboxyeocin is another good inhibitor, but it has to be loaded into the cell following esterification because the binding site is internal, and so it's not very practical. Around about 20 or so years ago, Ashok Grover developed a series of peptides that were based around the extracellular domains of the PMCA, and there was some hope that these might actually exhibit some selectivity for the different isoforms of PMCA. But these never really took off very well. The sort of IC50 values were in the sort of high tens of micromolar. However, my collaborators, Jean Bouton, Jérôme Leprince, and Karine Pouget from France, a couple of years ago, used these coloxin peptides as a structural backbone and developed a new generation of peptide inhibitors. They shortened the peptide sequence, they introduced non-natural amino acids, they chemically modified them to maintain that tertiary structure in an attempt to generate better, more improved, higher potency and potentially selective PMCA inhibitors. And they came up with a series of equipotent 5 residue-long analogues with low micromolar IC50s. And we have tested 3 of the most potent of these peptide inhibitors using our calcium clearance assay, and they are effective, particularly Cal1D80, which seems to flatline PMCA activity after a couple of minutes of initial clearance. We don't really understand how this works, but it definitely flatlines the calcium clearance. We've also tested these peptides on a cell migration assay. This is a gap closure assay where we load the cells into a chamber with a plastic insert. We remove the plastic insert and the cells migrate into this gap. And these are on BXPC3 cells, which have a very high migratory phenotype. Within 12 hours, the gap is completely closed, but then you can see that these different peptides are very effective at stopping migration. We've also used a cell invasion assay where we grow the cells on a layer of matrigel and then monitor the movement of the cells through this matrigel layer, and we can see the cells emerging on the underside of this transwell filter, and we can label them with Hirsch and then count the cells. And as you can see, these are preliminary data. We've not done that many experiments yet, but I think this suggests that these peptides are very effective at inhibiting cell invasion. So despite the preliminary nature of these data, this novel generation of peptide inhibitors of PMCA may represent potential antimetastatic drugs. And the next phase of our research is to generate pancreatic cancer cell lines that exclusively express each of the PMCA isoforms, where we can test all of these different peptide inhibitors to determine their potency and their selectivity, and then translate that to cancer hallmark responses and metastatic models of pancreatic cancer. In addition, I mentioned before that ATP drives the calcium pump, cellular energy, and cancer cells exhibit an altered metabolic phenotype. And the textbook view of metabolism is that most of the ATP comes from mitochondria, whereas a smaller amount comes from glycolysis. But cancer cells exhibit a phenomenon known as the Warburg effect, where there's a downregulation of mitochondrial metabolism and an upregulation of glycolytic metabolism. And this upregulation of glycolytic metabolism is very important because these glycolytic intermediates provide the building blocks for biosynthesis for rapidly dividing cells. In addition, lactic acid accumulates, and this is transported out of the cell by monocarboxylate transporters, and this extracellular acidification is very important for facilitating matrix metalloproteinases and cell invasion. Also, this acidity is important for inhibiting the immune surveillance, which is another important hallmark of cancer. So this upregulated glycolysis is very important for maintaining the phenotype of cancer cells, but ATP production remains important for maintaining ATP-dependent processes, including the PMCA, but also the sodium pump. That's another important one. And so we reasoned that cutting off this glycolytic ATP supply may render these cells more sensitive to cytotoxic calcium overload and cell death. And of course, that's what we found. So we measured metabolism using the Seahorse XF analyzer, which simultaneously measures oxygen consumption rate as a measure of mitochondrial metabolism and extracellular acidification rate as a measure of glycolysis. We also measured ATP using a FRET-based recombinant fluorescent reporter of ATP, which effectively changes color when ATP binds or dissociates. And so what these data show is that if we inhibit mitochondria with a ligamycin, which is an ATP synthase inhibitor, as you can see, the oxygen consumption rate drops, which is what you would expect. It's a mitochondrial inhibitor, but there is a compensatory increase in glycolysis, which maintains ATP constant. However, if we inhibit glycolysis with iodoacetate, this is a GAPDH inhibitor, you get a drop in ECAR, which is what you'd expect, but the compensatory increase in OCAR is insufficient to maintain cellular ATP, and therefore ATP goes down. Consequently, if we now measure PMCA activity, ligamycin does nothing, and iodoacetate inhibits the PMCA, and this leads to toxic calcium overload, whereas ligamycin does nothing. And we've repeated these experiments with a range of different metabolic inhibitors, and we've measured cell death, and this is just another example where we've used bromopyruvate as a glycolytic inhibitor. This is an inhibitor of hexakinase, and we've used the protonophore and mitochondrial uncoupler, CCCP, and this does nothing, okay? But the problem with this is that these are all quite dirty metabolic poisons, and these do not have any therapeutic value. And so the next phase was to try and understand which of the glycolytic enzymes were responsible for this upregulated glycolytic flux to fuel the PMCA. And PKM2, or pyruvate kinase M2, was found to be overexpressed in pancreatic tumors, and this led to poor patient survival. This is at the terminal end of glycolysis, is responsible for producing ATP and pyruvate. Also, another enzyme which is absolutely critical, which is PFK-FB3. Phosphofructokinase, fructose bisphosphatase 3. This is a maximally activated enzyme that produces the glycolytic intermediate fructose 2,6-bisphosphate. And if you remember from your early undergraduate biochemistry lectures, this is an allosteric activator of phosphofructokinase. This is the rate-limiting and irreversible step of glycolysis. And by activating this enzyme, it drives glycolytic flux to produce ATP. And also, PFKFB3 is overexpressed in pancreatic cancer and leads to poor patient survival. There are two other important enzymes here, PFKP, which is a PFK1, and PFKFB2, which is also overexpressed. And I'll come back to that when I talk about pancreatitis. We can inhibit PKM2 with a drug called chiconin, which is derived from traditional Chinese medicine, and it has around about a 20-fold higher activity against PKM2 versus PKM1. This inhibits cell proliferation as assessed using the sulforhodamine B assay. It induces a loss of cell viability as assessed using a tetrazolium-based assay. It induces cell death as assessed by PARP1 cleavage, and it inhibits cell migration. In addition, chiconin, as you would expect, inhibits glycolysis as assessed using the Seahorse EXIF analyzer. It causes a loss of ATP as assessed using luciferase assay. It inhibits the PMCA and it induces cytotoxic calcium overload. In addition, we can inhibit PFKFB3 with a drug called PFK15, and this also reduces cell proliferation, reduces cell viability, induces cell death. And what we've got here is we've combined, we've compared this to human pancreatic stellate cells, which are another cell type found in the tumor microenvironment, but these are not cancer cells. And the drug has less of an effect on these cells. But in cancer cells, this drug inhibits PMCA activity and it induces toxic calcium overload in myopathic cells, the cancer cells. But as you can see in human pancreatic stellate cells, there's no effect. And the reason for that is that human pancreatic stellate cells do not express PFKFB3. And this proves that the drug is acting at its target. So collectively, this is suggesting that targeting these two important glycolytic enzymes might be an important therapeutic strategy for treating pancreatic cancer. Okay, I'm now going to switch fields to pancreatitis. This is an inflammatory disease of pancreatic acinar cells. These are the cells that secrete digestive enzymes. The major causes are excessive alcohol consumption and fat consumption, which is particularly relevant at this conference, given that there's a bar that's open every night. And this leads to the accumulation of fatty acids and ethanol metabolites within the pancreas. Also, gallstones can cause pancreatitis, and this can lead to the reflux of bile acids that are toxic to the pancreas. And both bile acids and fatty acid ethanol metabolites converge on the mitochondria to inhibit mitochondrial function, leading to ATP depletion, inhibition of the PMCA, cytotoxic calcium overload, necrotic cell death, and then a cascade of local and then systemic inflammatory response, which are the hallmarks of acute pancreatitis. And what I hope I'm going to convince you is that, well, insulin can inhibit or block almost all of these different stages. There is a strong link between acute pancreatitis and diabetes, not least because pancreatitis can cause collateral injury to pancreatic beta cells, leading to a loss of insulin secretion. This is known as type 3C diabetes. There's also evidence that pre-existing diabetes can also make pancreatitis more severe. And the mechanism for this was poorly understood until we discovered that insulin has a direct protective effect on pancreatic acinar cells by preserving cellular ATP and maintaining a low resting calcium. Therefore, during the course of acute pancreatitis, the extent of collateral injury to the beta cells will lead to a loss of insulin secretion and a loss of this protective mechanism, leading to a much more exaggerated cellular injury, which then leads to a more severe acute pancreatitis. This slide summarizes basically the last 10 years of work that's summarized in this Nature Communications article. We have induced acute pancreatitis using two separate models. There's the cerulean hyperstimulation model. Cerulean is an analog of cholecystokinin. It induces a cytotoxic calcium overload and mimics the downstream pathology, but is not a very good etiologically relevant model, whereas palmitoleic acid and ethanol is a good etiological model of alcohol pancreatitis. And we've done both of these models on two separate transgenic mice, the type 1 diabetic Akita mouse, where insulin secretion is impaired, and the pancreatic acinar cell conditional insulin receptor knockout mouse. Here, the insulin receptor has been specifically deleted in pancreatic acinar cells using a tamoxifen-inducible Crelox gene deletion system. And this leads to a much more severe acute pancreatitis. And the reason for that is that insulin increases glycolytic energy supply to the PMCA. And this can be emphasized in cellular models of acute pancreatitis. So in this set of experiments, insulin prevents palmitoleic acid-induced ATP depletion, PMCA inhibition, cytotoxic calcium overload, and necrotic cell death. And all of these responses are prevented in acinar cells from Procerco mice that lack insulin receptors in the acinar cells, which is what you would expect. The mechanism is very similar to what we observe in cancer cells. There is a switch from mitochondria towards glycolysis, and we can assess this by measuring NADH autofluorescence. So a lot of NADH comes from the mitochondrial Krebs cycle. If we uncouple the mitochondria using this protonophore, CCCP, all of the NADH within the mitochondria is consumed, so you get a decrease in NADH autofluorescence. If we then inhibit GAPDH, which is a glycolytic source of NADH, you can get a further decrease. So we've effectively separated mitochondria from glycolytic metabolism. And then when we pre-treat with insulin, you can see a switch. There's a decrease in mitochondrial metabolism and an increase in glycolytic. And this is likely very important for fueling the PMCA during acute pancreatitis and providing protection. The molecular mechanism is also very similar to cancer cells because insulin activates AKT, and AKT directly phosphorylates the key glycolytic enzyme PFK-FB2. And again, it's very similar to PFK-FB3, except it's a tunable enzyme. And phosphorylation increases the activity of this enzyme, thereby driving glycolytic flux to produce ATP. So in other words, this mechanism acts as a volume control on glycolysis and preserves cellular ATP even in the face of impaired mitochondrial function. And so if we take a step back, you might come to the conclusion that insulin might be a good therapeutic strategy for treating acute pancreatitis, but that's not the case because severe acute pancreatitis patients are severely metabolically compromised. They have huge amounts of adrenaline, cortisol, glucagon elevated. They have elevated cytokines, which inhibit insulin sensitivity. So glucose control in these patients is very precarious and very dangerous. So we are now on a quest to find insulin mimetics that mimic this mechanism of activating PFK-FB2.